everyone, I'm Keith. I'm Bryson's dad and one of the founders and leads of GRIN. So when Bryson got his diagnosis a few years ago, we started thinking about him having a disease that we just thought of as GRIN1. And the more we learned about GRIN1 and the other genes and patients with the other genes, the more I came to realize that it was more of a broad disorder, that there were a lot of similarities between GRIN1 and the GRIN2B genes. And lately, Dr. Johannes, who we'll hear from shortly, has been convincing me to think of this as an even broader category of conditions, because in addition to GRIN, G-R-I-N, there's also G-R-I-A and GRIK and GRID genes that all code receptors that have similar form and structure to the NMDA receptor. So we were planning originally to start with Heather, who's a GRIA 4 mom, but she's having some trouble connecting. So instead, we're going to start with Dr. Johannes Lemke, who is going to tell us a little bit about the similarities and differences in this broad category of ion receptors.
I thought we were starting with Derek.
Oh, we're starting with Derek?
Yeah.
Okay.
Sure. Okay. Shall I go and share my screen then? Okay.
Yes, go ahead.
Okay. Perfect. Okay, so I'm hoping everyone can see this screen and everybody can hear me. Okay, since
we don't see your screen yet.
You don't see my screen. Why is that possible? I'm saying share, but it's not sharing. Let me try it again.
When you click share screen,
yeah,
what comes up?
It comes up with your share screen, and it says entire screen, share, and is this it?
Yes.
Okay, great. Sorry about that. Oops. Back again. Here we try. How is that? Can everybody see this?
That's perfect.
Okay. Okay, so maybe I can just start by thanking Keith and everyone for this kind invitation to do a little presentation for you today. I also want to thank my colleague Steve Janelles, I think, who put Keith and I in touch, and then Keith invited me to come along with the presentation. As my opening title suggests, I'm a professor at McGill University. I actually work in this building just across here. It's right in the heart of the biomedical research campus here at McGill. And I think the reason I'm here is to tell you something that maybe many of the people who have children and kids with GRIN disorders, which encode a protein called an NMD receptor. But that NMD receptor is part of this super family of proteins called ionotropic glutamate receptors, which you should all see, which are kind of summarized in this very kind of simple schematic presentation here. So you've been hearing, if you've been following all the sessions this morning, you've been hearing about NMDA type ionotropic glutamate receptors, but there's at least 2 or 3 other types of ionotropic glutamate receptors. I've only listed 2 of them here for simplicity. There's the AMPA type, which I'm going to tell a little bit more about in detail. And if Heather had been on the call, she would have told you a little bit about her son, who has a point mutation in one of these proteins that encode an AMPA receptor. And there's also a kainate type of ionotropic glutamate receptors. And something I've missed is there's an orphan class of glutamate receptors, which I've omitted just because of the time constraints that I have. So something that may not be immediately clear to many people who have been learning about GRIN and the conditions associated with it is that our knowledge about ionotropic glutamate receptors goes back at least 30-odd years. And during that time, we've actually learned that ionotropic glutamate receptors are involved in many, many different types of CNS disorders. And in fact, about in the early 90s, NMD receptors were linked to be important in a lot of the cellular death that occurs in stroke. And more recently, it's been tied into Alzheimer's disease. AMPA receptors similarly been tied into other kinds of neurological disease, including ALS, or motor neuron disease, and this very aggressive cancer. in KNF receptors has been tied into neuropathic pain mechanisms in epilepsy. The other thing, of course, that's become evident from the work from Dr. Lemke and other people who have been doing genetic analysis of glutamate receptors is that all of these different types of ionotropic glutamate receptors, point mutations in them, all give rise to neurodevelopmental disorders. And so if you're a researcher spending a lot of time thinking about ionotropic glutamate receptors, it's not exactly entirely surprising that glutamate receptors have such a very negative impact when things don't work with them very well. And the reason for that is that ionotropic glutamate receptors, they actually constitute the hardwiring of the brain. So in other words, when you're trying to build a brain, one of the key elements that you have to build into that sort of important architectural element is signaling mediated by ionotropic glutamate receptors. So therefore, if their signaling properties go awry, if any of these go awry, then it leads to a whole bunch of types of diseases. Now we're learning that they're all intricately involved in neurodevelopmental disorders. So if we can take you back a little bit in time, there was something that happened in the sort of early 90s as well, which I think has been really instrumental in for many of the people here who have come to this meeting to learn about the GRIN disorders. And that was really done by two major labs. There's a few other labs involved in it, but by Peter Seberg's lab in Germany and Steve Heinemann's lab at the Salk Institute in California. And what they often did is they cloned all the genes that encode the proteins, which are listed here, for the different families of ionotropic glutamate receptors. So I apologize for using the old nomenclature for NMD receptors, but you can see that they're made up of these different types of genes that come together to form the AMPA, NMDA, and KNF receptors. And so those observations, which were done, 30 odd years ago, were actually very important for many, many people listening to this presentation, right? For a couple of reasons. The first thing, of course, if you know what the gene is, then you can come up with genetic testing and then, of course, eventually diagnosis. And of course, that seems a very important aspect for patients and their parents, etc., in trying to understand what's happening to the child. The other thing, of course, and I don't know if we've really heard about it in great detail yet, but the work of Amy Ramsey, for example, who's been making transgenic mice with different types of mutations, that if we didn't have the genes, identify these genes, then of course we couldn't go off and make these transgenic mice and then look at their behavioral properties and their physiological properties. And then finally, if you have all these genes, then you can actually go off and make a huge amount of protein and look at structures. And so now we actually have full-length structures. This is one of an AMPA receptor, which was crystallized by Sasha Sobolewski and Eric Gould almost 10 years ago, in fact, right? So just that identification of the genes was incredibly important in our understanding of ionotropic glutamate receptors, and of course, leading it into clinical testing, etc. While all the biochemists were working, then the physiologists were also working away behind the scenes as well. And what we realized is that when you look at AMPA and NMD receptors, and you look at these very, very small connections between one nerve cell in the brain and another nerve cell, which has the AMPA and NMD receptors in them, is that there's an incredible interaction between them. They basically work as partners with each other in the brain. So just to give you an example, in a developing brain, it's really the NMD receptor that turns up first, and then it starts to bring the AMPA receptors in. So what that means is that this very important partnership, without getting into all the details of it, this partnership is actually critical for brain development. And I believe that's the reason why many of you are coming to this symposium to try to understand what kind of aspects of brain development are being affected by these single point mutations in some of these proteins that we're talking about. But not only is it important in brain development, of course, is that this interaction between the NMD receptors and the AMPA receptors is also critical for the adult brain, because that's how we lay down memory circuits, is by an interaction with these. So now what I'm going to do is tell you a little bit about the AMPA receptors, which I've spent quite a bit of time looking at. And of course, Heather's son, Max, the point mutation is actually found in the AMPA receptor of Max. And so the challenge with AMPA receptors is that they not only do they have these 4 genes that make up AMPA receptors, but they have all of these different families of helper proteins which bind to AMPA receptors, bring them into the neuron and put them in the right place, and also affect their function. And we spend quite a bit of time trying to understand that. And so you can see that what's emerging is this very, very complex picture between the AMPN and NMD receptors actually working together. And then you've got this extra layer of complication of all these other proteins which are there. The advantage of that now is, now that we're starting to have a fuller picture, what we're realizing is there's another level of complication on top of this. So here's a full-end structure of an AMPA receptor here in the green and gray. And here's one of these helper proteins. You see there's two on this particular AMPA receptor. But in some cell types, and this is something that we reported only back 1 year ago, is sometimes they have 4 helper proteins, and then sometimes they have 2. So when one is starting to think about therapy, thinking about pharmacology and all these kind of things, then you have to keep this in mind, okay? When you're starting to think about this disease and in the context of Max, which hopefully you're going to hear about from Heather, in the context of his disease, then you have to start to think about all these extra levels of complication into it. But the great advantage of working these things out is now, with the help of geneticists who have been now pinpointing different mutations. So what I've done here is got a structure of an AMPA receptor, which is shown here, and here's another type of auxiliary protein, which is found here. And what I've done is I've identified in yellow or in green single point mutations which are found. Some of them are published, some of them are not unpublished from work that we're doing, that are within the AMPA receptor pore forming or the core part of the protein, but also some of them which are on the auxiliary subunits, these helper proteins which are there. So what it's really saying to you is that the AMPA receptor signaling complex, just like the NMD receptor complex, is being pinpointed by all these different types of mutations, which are either affecting the functionality of the receptor or the signaling protein, or its ability to get on the surface of the cell and then act in a signaling capacity there. So what emerges from that, and what you may be thinking right now, is this sounds all incredibly complicated, and there's no way, it's going to be very difficult to try and come up with some kind of therapy. So what I wanted to do in sort of finishing this very, very brief little conversation, I hope it hasn't been too detailed in terms of the science, is just finishing on something which is to give you more of a kind of philosophical perspective of how, at least how my lab is starting to think about it. So don't worry about all the details which are on this slide. What I really want to point out to you is that the NMD receptor and the AMPA receptors, so these sites which are called glutamatergic synapses, they're kind of at the heart of this rather complicated and interrelated system, which is critical in all neural circuits within the brain. The NMD receptors, for example, regulate how excited neurons get. They also affect how they get inhibited. So if you have defective NMD receptor signaling, you don't get the right inhibition. And so that could easily lead to epilepsy, for example, that you see a lot with GRIN mutations. You also interact with astrocytes, and together, this signaling actually affects blood flow into the brain. And that's something that most people haven't really been thinking about, but that's something that's pretty well established for glutamatergic synapses, that they're also affecting the blood flow into the brain. So you can imagine, if you look at this in a more holistic sense, you can see that if you have point mutations in a sodium channel or NMD receptor or AMPA receptor, that they will all eventually impact this rather large bit of physiology, so that in the end, you have some sort of kind of defect that may play out in neurodevelopmental disorders. So just in finishing, I hope I haven't been too fast in trying to take you through some rather complicated things. If anybody's interested in learning more about this, feel free to go to my website. There's the contact information for me, my email, telephone number. Please feel free to contact me if there's something that you think you'd like to learn a little bit more about. And likewise for the researchers, feel free to reach out. I'm happy to tell you a little bit more about AMPA receptors and how we think they interrelate with NMD receptors. So with that, I'd like to thank again Keith and of course Steve for making the connection. And for all of you for your attention, I'll be very happy to take some questions. So thanks again.
Thank you, Dr. Bowie. And now I'd like to introduce Dr. Johannes Lemke from University of Leipzig in Germany.
Yeah, thank you very much. My presentation is not shareable, so someone is setting it up right now. I only have 2 slides only, so
no problem. Yeah, it looks like Heather's still having problems connecting. But in the meantime, maybe I'll ask you a question, Dr. Bowie. That I mean, it seems like we have, both GRIN and GRIA. These individuals have rare diseases that focus specifically on this gene, but you're suggesting that there could be maybe dozens of other neurological diseases where there might be another cause, but it all revolves around the GRIN and GRIA genes potentially, and the NMDA and AMPA receptors.
Is that towards me?
Towards you, yeah.
Yeah. So I'll give you an example. There is a mutation in the sodium channel, which maybe many people on this podcast won't appreciate. So in 1.2, it was published last year, and what they find is that even though it's affecting a protein that seems quite distinct from glutamatergic signaling, NMDA receptors and AMPA receptors, even though that's the case, then you do get deficits, the kind of deficits that you see in some of these patients with GRIN disorders. And so what I'm trying to say is there's a big soup whereby all of these molecules are actually signaling to each other to tell each other, I'm over-signaling, under-signaling. And when there's a defect in one of them, especially the NMD receptor, it has this knock-on effect. So that some of the symptoms that patients are seeing, for example, of epilepsy, may not be directly related to signaling by the NMD receptor, but because the NMD receptor isn't signaling, for example, to a GABA receptor, to actually bring about inhibition in the brain. And without that inhibition, you get epileptic seizures. So that's what I'm trying to say. It's a slightly different approach from focusing on the mutations and trying to potentially, they're kind of complementary. It's not that one is better than the other.
Right.
We're trying to see it in this more holistic sense as a physiologist.
Thank you.
Yeah.
So I don't know if we have your slides available. Clarissa, are you able to start them up, or should Johannes begin?
Otherwise, I just talk without slides. Yeah, so actually, in my first slide, I had an image of the synapse showing you that they're actually, there it is.
There it is.
Great. Yeah, so that's actually illustrating a crucial part, or the synapse, which is crucial for the neurotransmission and the interaction and signal transduction between neurons. And what I marked there in red in the middle are the two receptors, NMDA and AMPA receptors, that interact with each other and that share a lot of similarities. And why I think this image is quite interesting is all these proteins that are marked in there are ion channels and receptors and synapse proteins. And it doesn't matter where you have a genetic variant, you will end up with a disorder which leads to developmental delay, intellectual disability, and likely to epilepsy. And what is also marked there is all the drugs that are being applied currently, anti-epileptic drugs. And if we know which protein, which receptor, which ion channel in the brain is not working well, we may know better which drugs to use. So there are some specific drugs that target the NMDA receptor, as most of you know. And there are also some drugs that target the AMPA receptor. And when we identify a pathogenic variant in a patient and know that it is a GRIN1 patient or a GRIN2B or a GRIA4 or whatever, then we hopefully will be able one day, maybe not right now yet, to give these patients drugs that are specifically targeting their genetic defect and will help them much better than other standard drugs. And in the next slide, can you go one further, please? One slide further, please. Anyway, in the very little slide below there, I listed you the genes that are associated or that are known of these different gene families. So you all know that there are GRIN1, GRIN2A, GRIN2B, and GRIN2D so far associated with human phenotypes. All of them are developmental and epileptic encephalopathies. GRIN2A is the only gene where individuals also can have milder phenotypes and may not be intellectually disabled. but GRIN1, 2B, and 2D, they all have at least mild, but most likely severe or profound intellectual disability. And if we look at GRIA, then there is a family of a few genes, and GRIA2, GRIA3, and GRIA4 are also associated with disorders that are going along with developmental delay and with intellectual disability and also seizures. So it's the same spectrum of developmental and epileptic encephalopathies. And if you look at the GRIT and the GRIK genes, there's not much known yet. So there are not many human diseases that are linked to these genes in terms of pathogenic variants are causative for the respective disorder. That's why I marked them in gray. But they also are contributing to the same phenotypic spectrum of developmental and epileptic encephalopathies. GRIT2 mutations are the only ones that are labeled, I would say, with a different term. They are named spinocerebellar ataxia. But actually, if you look at the phenotype of these rare patients, then you still see similarities to the other developmental and epileptic encephalopathies. So the main and leading symptoms of all the GRI disorders, no matter of GRIN or GRIA or GRIK or GRIT, is actually intellectual disability and developmental delay, seizures, muscular hypotonia, so a muscle weakness, movement disorders or ataxia, and as the phenotype overlaps in all these disorders, as well as the domains and the structure and the morphology of the receptor, I think it would be worth to lump them together in one pot. And I believe as all these receptors or as mutations in these receptors mediate either loss or gain of function, and that's a similar pattern in all of the subreceptors, I think that it is scientific progress that is made in one of the receptors will also help the other receptor families. So we can learn from each other. So that's why I think we should just join forces among researchers and among parents to put these 4 receptors together.
Thank you so much, Dr. Lemke. So it looks like, unfortunately, it looks like Heather is not going to be able to share her story today. So we have time for maybe 1 or 2 questions. So I see one question here, which is maybe for Dr. Lemke, but the question is, are there known variants in GRIK or GRID, GRIK or GRID genes that are disease causing?
Yes, for GRID2 and for GRIK2, they are disorders that are associated with, or they're variants that are associated with the human phenotype.
Okay, and so just a reminder to anyone, if you have any other questions, you can put them there in the Q&A. But I did, have talked to Heather, and her story is very similar to many of the other stories that we've heard from GRIN patients. I think, if you heard it, you would not know that she was not that her son Max does not have a GRIN disorder. And so we've actually, at Cure GRIN, we've taken some steps to work more closely and look more closely into these other genes. And so Heather has joined our board of directors in an informal non-voting capacity so that she can learn more about what we're doing, and we can learn more about GRIA genes. And we have plans to start broadening out and doing some other things related to those genes as well. Some of these families are going to be doing fundraising for us over the next few weeks, and so the funds that are raised for these other genes, we're going to invest in research related to those genes as well. So we're almost out of time. I really want to thank both of you for your presentations, and to let everybody know, we have a short break coming up after this session, and then at 1:30, so in about 15 minutes, the next series of sessions will start. So there's a session on gain of function and loss of function, what they are, why that's so important to know. There's a session on ketosis as a potential treatment for epilepsy, and maybe as a treatment for other potential conditions related to GRIN. And then there's also a sibling chat. This one is for kids, so for 12 and unders, can join with other siblings, and it's a session led by teenagers and adults with GRIN, and it's a place to go and just chat in a safe place, share your story, ask questions of each other. So hopefully everybody is having a good day at this session, and we'll see you all in 15 minutes. Thank you.